Well, now that we understand a bit about what the physical structure of DNA is, I thought I would just give some examples of potential test questions that you might see, are questions that you might see in the book, and how to maybe to approach them. Very straightforward. Again, there are a number of problems in the book, and easily searching the web, you can see lots of examples of problems. But I just thought I would just do a few here. Um, First, just a few keywords, just things to remember. Complementarity is important. A to T and G to C. Remember how many bonds are between A and T is 2. G and C is going to be 3. So you need to think about that. The words anti-parallel should now mean something to you. 5 prime, 3 prime versus 3 prime, 5 prime. That's going to be important for not only these problems, but also when we do replication, as well as when we do transcription. We'll need to know which direction we're working from. So those points will become very important in problems. And then this idea of polarity that goes along with that. Which of the free groups were we going to add or groups to it? So the 3 prime OH group down on the number 3 carbon, and then the 5 prime OH group that's found off the POH group that's found off of the number 5 carbon. That's going to be your phosphodiester bond region. Okay, so we want to work with those. All right, so let's just try a few straightforward problems here. First, let's take something that maybe a bunch of words, we've just extracted out the wording, but a molecule with 5,000 base pairs. BPs are often abbreviation for base pairs. So we mean here, when you see base pairs, you should know that you're dealing with double-stranded DNA, right? And be careful, be sure whether it says, just 5,000 nucleotides, that's not necessarily double-stranded. So you have to think through that part. But this part's pretty clear. We talked about 5,000 base pairs. All right, so some questions. How many nucleotides is this? Well, it's obviously 10,000, right? Because you've got base pairs. A pair is 2. So 2 times 5,000 is 10,000. And that's the double-stranded part I was talking about, okay? So just be careful, read that part of the question. Then you might be asked how many sugars. Well, just think about what a nucleotide is. It's a phosphate group. It is a base that you have added on ATGC, and then there's a sugar for each one. So for each one of these nucleotides, you're going to have a sugar. So the answer there is 10,000. Okay. And then we go back to the Watson-Crick model, thinking about things like distances and turns. We said with the B-type DNA, beta, that they described, there's about 10 bases per turn. So how many forms do you have? Well, if you got 10 bases per turn, okay, and you've got 5,000 bases here, all right, so you're looking at about, what, 10 divided into 5,000 will give it approximately 500 turns. Okay, it's actually a few less than that because it's about 10.5 bases, but that's fine. Okay, that is close enough for what we're going to do. So we'd go around about 500 times in 5,000 base pairs. All right, what's the total length? Well, we know how far the steps are apart in beta, right? It's 0.34 nanometers. So if we were to take that information, we say it's about approximately 3.4 nanometers per base pair. All right, there's 5,000 base pairs, so we've got about 1,700 nanometers would be the length of this. Now, just to make a point, if you really wanted to do this accurately, you would actually use 4,999. You would subtract one of the steps. I mean, if you're thinking about it, you're counting these inner spaces, and this top one, you're already there. So you don't count that one. It's always 1 minus that. So if you're actually going to do this, it would be 4,999 times 0.34. But I think, what is that going to be, about 1,699 point something? So, it certainly rounds up to that. But just be careful and when you're working problems that you do, particularly if it's only a few bases, that you use the correct number. And then you can always throw anything at this. You can ask yourself, or you can have your study partner say, okay, how many phosphodiester bonds do we have? In other words, there's got to be one between here and here, one between there and there, and one between there and there. Count them up. Well, you got double-stranded DNA, though, so you have to think about it from both sides. So how much do we have? Well, certainly not 10,000, okay? There's some numbers slightly smaller than that we actually have. So just think through that. It's the same way with the steps. You have to think about, okay, there's 1, there's 2, there's 3, okay, but it got to stop up here somewhere that I don't count another one above it, right? Okay. All right, so what about some other things? Classic problem is we give you piece of double-stranded DNA, given, and he would say, okay, how much G is there, or how much T is there, or whatever. The point is, since there's only 4 bases, if you know in their complementarity, if you know one, the percentage of one, you can figure out the other 3, no matter what they are, right? So if you know that there's 10% in this case G, right? How much? What else have we got, right? Well, how much T is there? Well, G and T don't link, right? There's no direct correlation. We know that. But we know how much G is there and it's double stranded. What we then know is how much C we have, right? If we follow Chargaff's rule, we know that these are complementarity. So we know there's an equal amount of C, okay? And then we can say, okay, the answer is 40%. Why? Because if these two add up to 20%, then there must be 80% left. That 80% has to be divided between A and T. And so the answer must be, in fact, that we have this 40%, right? So how much of T is present is 40% of it. Okay? You do the same thing. You can do it in any model you want to make up. There's 22% A, how much C is there? 22% A, how much G is, whatever. As long as it's double-stranded, you can do it. If it's single-stranded, you have no idea, right? Because single-stranded, unless there's other types of information, there's no complementarity in single-stranded, so you can't do that, right? And maybe just a thought question. Okay, so we know that we've got 40% of T, right, or something like that. We could easily change this to a question like this. All right, now if you have a region of double-stranded DNA that's, say, 100 base pairs long, or 95 base pairs long, or 2,000, whatever number, how many hydrogen bonds do you actually have in that double-stranded region? Well, if you know the amount of G and C, right, then you would multiply that times 3, because there's 3 bonds between G and C. And then if you know the percentage that is A or T, then you would multiply it times 2 bonds, right? So in our case, 80% of it is A and T, right? Because 40 plus 40. So 80 times, and it's 100, so 80 base pairs are going to be AT, so that's 80 times 2. 20% is going to be GC, so that's 20 in this case times 2 base pairs, right? And just add those up. That would tell, or excuse me, 2 bonds. Add those up, and they'll tell you how many hydrogen bonds you actually have, right? So you can do that. You can do it with any number. We could throw 512 in here or whatever. Just figure out the percentages and work from there. So again, we're taking a fairly simple question, fairly common, that you find in just about any basic genetics book. We talk about DNA structure. And we've just modified it slightly to ask a different question. But once again, going right back to those Watson-Crick models, the whole point being that, you don't have just memorizing that stuff. It's not going to be enough. You need to be able to think a little bit about how to use it, right? And then our last little question, very classic questions. Given some information, is this double-stranded or single-stranded? There's a number of different ways to do this. Obviously, you could use the Chargaff's rules to do this. Sum of the purines has to equal the sum of the pyrimidines to be double-stranded. So A plus G, okay, is clearly not equal to this, so it's not double-stranded. In fact, you can just look at it and see that it's not double-stranded, because if it were, A should be approximately equal to T, and they're not close, and G should be approximately equal to C. Although they're fairly close, still, I would guess that this is not a double-stranded, and certainly by using one of Chargaff's rules, it's pretty clear that they're not double-stranded, right? And so you could then say, well, all right, well, this percentage is like this. Now, other questions that come up, survey sent to Mars, comes back and finds some new genomic material, and it's got U down here, some percentage of U, but no T. What is it? Well, it's RNA. Okay, well, is it double-stranded or single-stranded RNA? So you have to look at the percentages that go there, okay? All right, now, the next question might be about thermostability. Remember the basic rule. GCs have 3 bonds, ATs have 2 bonds, so things that are more thermally stable, will melt slowest, are actually those things that have the most GCs, and so ideally you might go out to one of these thermal vents in Yellowstone or somewhere where there's volcanic activity. Go to Iceland where they have lots of nice hot springs. What type of life? This red stuff here is a bloom of life. What's in there? Has it got a lot of GCs or has it got a lot of ATs? Which one is it going to have? So this one, we're just ordering these 2 stretches of DNA. Well, first you got to figure out if they're the same length, okay? And ours happen to be, I think, 10 bases, 1, 2, 3, 4, 5, 6, 7, 8, 10 both. They could be 12 and 14, so you need to count them first, okay? And then you just start looking for the G's and C's. So in the first one, we just have 2 GCs connections. On this one, we have 4, 1, 2, 3, 4 spots, okay? So in this case, it's going to be 2 out of the total length, and this is going to be 4 out of whatever the total length is. Well, our total length is fairly easy here, okay? I said it was 10, but looks like it's 9, sorry. So that's about 0.22 or about 22% GC that's found here. This one, 1, 2, 3, 4, 5, 6, 7, 8, 9, also 9, okay, not 10 as I thought. You've got 4 out of 9 here, okay, well that's going to be about 44%, right? So of these, if you rank them, this would be the most stable, number 1. This would be the less stable fragment right here. And if you ask the question, which one of these two would more likely be found in this hot spring, it would be the second one down here. It's very straightforward, okay? All right. That's really the major point of all this. That's working some problems. I would also like to point out fairly quickly that you need to remember and think about the differences between RNA and DNA, right? Just think about the forms. As far as function, DNA is primarily for genotype. It's the coding of the genetic information. RNA has lots of different things. It's often an intermediate for a phenotype, from going from DNA, the genotype to the phenotype. It can be genotype. It can be, you can have double-stranded RNA and it be genotype in some organisms. And it plays all kinds of other roles. It turns out it's going to be parts of the system for when we go splice out systems, we'll have RNA there. It's part of ribosomes. It has all sorts of jobs, usually where it's in a single-stranded form. But as far as what's going to be the genome, that's usually primarily going to be DNA, okay? Number of bases, well, it's the same, okay? It's A, T, G, C, so 4 bases here. And there are 4 bases in RNA, but you've now replaced the T with uracil, okay? The sugar is 2 deoxyribose, so we've taken off an oxygen off the number 2 carbon. And then we have ribose over here, so that you need to be able to look and tell the difference between those two. Just to remind you. Okay, the unit structure here, of course, is a nucleotide, and for RNA, it's the same. It's the same type of structure, so they do not differ at that point. And then the growth structure, DNA is almost always double helix. Okay, it's almost double-stranded, double helical form, unless it's being pulled apart for some reason. But certainly, as a genomic unit, it's double-stranded. RNA is generally single-stranded and linear. Okay, so it's not a helix at all, it's some kind of linear form. But when it's a genotype, as it sometimes is, then it does take a helical form. Okay, so in some organisms, the few organisms that have RNA as their DNA, if you will, it's usually a double-stranded helical form. Okay, so just some things to remember, and these can become questions as well that you may see on problems or working on tests. All right, now that concludes everything we want to talk about building DNA, kind of just bring back to you what it looks like and the structure that it has, and some of the rules that apply as far as model of DNA that we view as currently existing. And now we'll move on to some other topics that relate back to this. Primarily, we're going to talk about replication and some other things.